we discussed in our regular class about gametogenesis on the blackboard. Now see here, gametogenesis, it is a process by which diploid or haploid cell undergoes cell division and differentiation to form mature haploid gamete. It means formation of gamete, male gamete and female gamete. Right? So there are two processes in gametogenesis. One for male gamete formation, that is spermatogenesis, and second one for female gamete formation, that is oogenesis. So see, first here spermatogenesis means formation of a sperm by testis in male, that is male gametes, and oogenesis means formation of ovum by ovary in female, that is female gamete. Sperm and ovum are highly specialized sex cells, and gonads are mesodermal inverted, but gametes derived from endoderm. Sometimes this question asked that gamete derived from endoderm, right? See, there is a structure of testis. You people know this structure, right? So testis, and then testis, there is a here upper side, that is epididymis, and when you elaborate this further, one part, then you will see this. So here interstitial cell that produces testosterone. Here spermatogonia divided and produces spermatocytes. Here this see that there is Sertoli cell that help in nourishing the spermatozoa, and spermatozoa developing spermatozoa that we discussed, that type A, type B, then how spermatids, and then how spermatozoa, right? So this is structure just to understand. See what is happening in spermatogenesis. First stage, an outer layer of germ cell called spermatogonia. Then second stage, what is happening? Spermatogonia enlarges and form primary spermatocytes. Then what happens? That is most important at third stage. That each spermatocyte carried out first division of meiosis start here, but halt at, I already explained that. Which stage? Just speak it out. Which stage? Meiosis 1. Prophase 1 and diprotein stage, right? That is halted out, and after that, see here, there is each primary spermatocyte develop into secondary spermatocyte. So each secondary spermatocyte carries out second division meiosis to produce two spermatids, and finally there is a formation of a spermatozoa. This is more important to understand. What is happening? See, spermatogenesis, spermatogonium divide by the process of mitosis. Spermatogonium divide the process of meiosis, and that form primary spermatocyte, right? Then what happens is that primary spermatocyte prepare, enlarge, and then participate in meiosis process. So here see that first meiosis process, two steps, meiosis 1 and meiosis 2. So first meiosis 1, and meiosis 1, what is happening that there is a formation of secondary spermatocyte, right? There is a formation of secondary spermatocyte, and then secondary spermatocyte hunted at a place. I will explain, and then secondary spermatocyte develop into a spermatid, and further by spermatozoa, right? See here, what is happening that this is growth spermatogonia to primary spermatocyte. That is growth phase. That is growth phase. So after that first meiotic division, and then completion of first meiotic division, there is secondary spermatocyte. Right? And then the secondary division when completed, then formation of what? Spermatid, and finally spermatozoa. 2 points important to remember. primary spermatocyte halted at meiosis 1, prophase 1 diploid stage. Secondary spermatocyte halted at meiosis 2, metaphase 2 stage. That is very important to remember, right? Then further, see that this same thing. Mitosis produces more spermatogomia. Some return to basement membrane. So see that जब प्राइमरी स्पर्मेटोसाइट फिर मेयोसिस 1 कब शुरू करेगा, क्योंकि मेयोसिस 1, प्रोफेज 1, डिप्लोइडेस 1 स्टेज में हॉल्टेड है, तो जब एफएसएच स्टिमुलेट करेगा, जब फॉलिकल स्टिमुलेटिंग हार्मोन स्टिमुलेट करेगा, देन प्रोफेज 1 का डिप्लोइड स्टेज आगे जाएगा, और जब प्रोफेज 1 का डिप्लोइड स्टेज आगे जाता है, देन मेयोसिस 1 कंप्लीट होता है. सो सी दैट व्हाट इज हैपनिंग, दैट मेयोसिस 1 कंप्लीटेड एंड देन फॉर्म सेकेंडरी स्पर्मेटोसाइट. सो एफएसएच स्टिमुलेट मेयोसिस एंड स्पर्मेटोसाइट डिवाइड बाय मेयोसिस. Luteinizing hormone stimulates testosterone, testosterone stimulates meiosis 2. So meiosis 2 को भी complete करने के लिए क्या चाहिए? There is a need of testosterone, right? There is a need of testosterone. Okay? So see that, and again secondary spermatocyte divided into spermatids and then sperm cell. So यहाँ जो points लिखे हुए हैं उसको याद रखना बहुत जरूरी है, क्योंकि ये पॉइंट्स कॉम्पिटिटिव एग्जामिनेशन में पूछा जाता है, राइट? अब इसमें क्या क्या इंपॉर्टेंट पॉइंट है? एफएसएच मेयोसिस 1 को स्टिमुलेट करता है, राइट? एलएच टेस्टोस्टेरोन सीक्रेट करता आता है, और टेस्टोस्टेरोन जो है मेयोसिस 2 को स्टिमुलेट करता है, राइट? Spermatids differentiate into sperm by tail development and midsection and mitochondria etc. formation, right? Yeah, already we have discussed all those things. So see, what is spermatogenesis? So the process development of spermatids from the male primordial germ cells and their differentiation into spermatozoa under stimulation anterior pituitary gonadotropin hormone. Then primordial germ cell develop into spermatogonia. There are two types of spermatogonia, type A and type B spermatogonia. Type A that divide by mitosis to provide a continuous reserve of type B, and type B that enter spermatogenesis. Then what is happening that these occur after puberty and they remain in the wall of the seminiferous tubules. Spermatogonia form primary spermatocyte. They remain in the prophase of first meiotic division for 16 days. Each contain 22 pairs of autosomes and 1 pair of sex chromosome XY. Then divide into 2 secondary spermatocytes, that is meiotic division completed. Each secondary spermatocyte has equal chromosome, 22 autosome and X or Y sex chromosome. Each of these divide again second meiotic division, thus forming 4 spermatids. Further, each containing equal cytoplasm and haploid chromosome, 2 with 22 autosome and X and 2 with 22 autosome and Y. So see here, testes, there is formation of spermatogonia, then primary spermatocyte, then first meiotic division, then here see that there is formation of secondary spermatocyte. Secondary spermatocyte again form here. There is 4 spermatozoa, spermatids, and spermatids, spermatogenesis process, and then formation of 4 sperm. So this is normal process for formation of male gamete. Clear? See, from spermatids to spermatozoa, this is not easy process. I already explained that what is happening. So the process of all those things is for the motility of a sperm, right? So what happens? The newly formed spermatogenesis is almost perfectly spherical and has well-developed Golgi apparatus. Then what is happening? Then a small vesicle of the Golgi fluid gives rise to larger secretory granules called proacrozomic granules. The centriole start to migrate to the position beneath the nucleus, and that is opposite side the acrozomic vesicle. And third step, see that vesicle fusion continue until a large site acrozomic vesicle is formed, and that contains a dense acrozomic granules. The proximal centriole will give rise to the attachment point of the tail. The distal centriole will give rise to developing axony and inside the cytoplasm of the spermatoid. So this is a process, and finally what is happening? That see that there is a formation of cast cell, then acrozomal phase, then maturation phase, and finally see this structure form. So here go acrozome, that is more important, right? For penetration, because acrozome contains enzymes, right? See, this is haploid nucleus, right? Here that is junction. This is centriole, and from here there is structure that is tail. And initial stage there is helical mitochondria. See that helical mitochondria, and the element is 9+2. So just go and read the slide. The structure of mature sperm. This is the structure of mature sperm. See, acrozome contains enzymes which can digest the zona pellucida, right? Nucleus, that is haploid, and midpiece possesses helical mitochondria which provide the ATP for swimming and other process, and tail flagella that contains protein, fiber, and microtubule to strengthen and allow the tail to move respectively. Right? So this is mature male gamete. Now this is ready for the fertilization. Now see, again, what is happening? If the process will continue, then there will be problem. That's why there is a check and balance in the process, right? So what is happening that you people know that hypothalamus secrete gonadotropin releasing hormone, stimulating the anterior pituitary to release gonadotropin, that is luteinizing hormone and follicle stimulating hormone. LH stimulates the Leydig cell to produce androgen, and FSH stimulates the seminiferous tubule to undergo spermatogenesis. Then what is happening again? Under the influence of FSH, Sertoli cells secrete inhibin. Here see that under the influence of FSH here. Under the influence of FSH, right? Sertoli cell, here Sertoli cell secrete two things. One, inhibin, another one, ABP, androgen binding protein, and third one, Mullerian duct inhibiting substance. Right? So what is happening that? Inhibit again for the production of FSH. So in this way it is acting like a feedback mechanism. Right? So inhibin follicle stimulating हार्मोन को इनहिबिट करता है, साथ साथ ये गोनाडोट्रोपिन रिलीज हार्मोन को भी इनहिबिट करता है, right? Second one, what is happening? The second one, that is ABP, androgen binding protein, right? So ABP binds with androgen, increases its concentration around the seminiferous. यहाँ देखो, ABP androgen से bind करता है, that is testosterone से bind करता है और उसके यहाँ पर इसके concentration को बढ़ाता है. And third one, that is Mullerian duct inhibiting substance. So Mullerian duct inhibiting substance MIS causes the degeneration of Mullerian duct in male during fetal life, which otherwise develop into oviduct. For development of oviduct, Mullerian duct in future develop into oviduct. So in fetus, what is happening that this MIS, Mullerian duct inhibiting substance, where the MIS is हो सकता है इसमें नहीं दिया हुआ उस लाइन में. So MIS का काम क्या है कि MIS को जो मुलेरियन डक्ट है उसको इनहिबिट कर देगा, जिसके वजह से जो फीमेल में ओविडक्ट बनता है ओविडक्ट ना बने। So male की डेवलपमेंट में हेल्प हुआ ना। तो in this way see that you three things remember that inhibin, ABP and MIS play very vital role for the spermatogenesis. Clear? Now yes, just stop. जो अभी आप लोग पहले में क्लास में पढ़ाया था उसमें कुछ मिस्टेक था। Try to understand that. Here see that spermatogonia के बाद ये परमानेंट spermatocyte बना, right? And then meiosis 1 स्टेज में ये रुक जाता है। meiosis 1 के prophet 1 डिप्लोइड नहीं, meiosis 1 में हॉल्ट होता है। किस स्टेज में हॉल्ट होता है नहीं मालूम।